Salam sejahtera. My name is Binti Zaharin. My matric card number is AN180026. So today I will be presenting my final year project with the title of The Pollutant Removals and Loading Capacity by Microalgae Botryococcus SP Alginate Beads in Greywater Treatment. So firstly, my supervisor's name is Prof. Madya Dr. Nur Shuhaila binti Muhammad Sunar, while my co-supervisor's name is Dr. Najiha binti Muhammad Apandi. So firstly, the purpose of this study conducted is because there are some problems that appear, which one of them is the conventional greywater treatment is not a sustainable approach because of the chemical additives that were added to the treatment. Furthermore, the bathroom greywater is rich with nutrients such as nitrogen and phosphorus that came from shower gel, shampoos, conditioners that may have high concentration of nutrients. Thus, the method of this study is to use the microalgae as biological treatment in greywater treatment, or can be called phycoremediation because it is proven to be uh, a simple, efficient, and environmental friendly method. So the solution for these issues are by using phycoremediation process. So the microalgae that were used for the phycoremediation process is Microalgae Botryococcus sp Alginate Beads. There will be various loading capacity of the microalgae alginate beads from 200 gram to 800 gram. The purpose of using the different loading capacity of microalgae beads is to know the efficiency of parameter reduction and nutrient removal in bathroom greywater. Next, the absorption of maximum loading capacity of the Botryococcus alginate beads will be done with the use of pseudo-second order kinetic model. By the end of this study, uh, it can be concluded that 600 gram of loading capacity is the optimum value for the parameter reduction and nutrient removal in bathroom greywater. So firstly, why phycoremediation is used in this study? It is because it has economical potential as well as environmental sustainability. Furthermore, the phycoremediation process is widely available and simple. Lastly, it has the ability to remove nutrients such as nitrogen and phosphorus in wastewater uh, as, as well as greywater. So, why Botryococcus sp alginate beads is used in this study? It is because the microalgae has high efficiency of nutrients removal in bathroom greywater as proven in some journals. Next, Botryococcus sp is rich in lipids and provides an environmentally acceptable approach, which they do not use any chemicals. Next, the efficiency of using phycoremediation as a method to treat greywater and wastewater has been proven by previous journals and research, which some of them is by using Botryococcus sp in bathroom greywater, they have removed, they have reduced the biochemical oxygen demand concentration as well as chemical oxygen demand concentration by 95 and 88% respectively. Next, Botryococcus sp is used to treat the industrial wastewater where it removes total phosphorus and total organic carbon by 85.5 and 87.2% respectively. There are some other microalgae that were used other than Botryococcus, which is Sandesmus sp, um, which the microalgae use the wet market wastewater to remove the total nitrogen and total organic carbon by 92.8% and 85% respectively by previous research. So I will move on to the methodology of this study. From this poster, it can be seen that the flowchart of this study. So the microalgae that has been harvested using flocculation process will be immobilized using calcium chloride solution, and it will be shaped into the bead shape. So next, the sampling of bathroom greywater. It was taken at the residential house at Kampung Melayu Raya Sagil, Tangkak, along with the sampling of bathroom greywater. The characteristic of bathroom greywater will be checked, such as the pH, the turbidity of the bathroom greywater, the initial concentration of biochemical oxygen demand, the initial concentration of chemical oxygen demand, as well as the initial concentration of total phosphorus and total nitrogen. Next, the phycoremediation process was done for 14 days with the interval of 2 days to get the analysis for each parameters and nutrient. During the phycoremediation process, the parameters concentration and nutrients concentration analysis will be done. The data was took each 2 days for 14 days. The Microalgae Botryococcus sp alginate beads was also analyzed in this study. Next, the experimental data will be compared with the kinetic model, which is the pseudo second order kinetic model, to see if the experimental data match with the kinetic model pattern. Next, I will move to the results and discussion. Okay, there are 4 graphs which combine the experimental data and pseudo second order kinetic model. The scatter graph shows the experimental data, while the line data shows the pseudo second order kinetic model. So as we can see from the graph, it can be seen that the phycoremediation process using Microalgae Botryococcus sp alginate beads is efficiency for all parameters reduction, which are biochemical oxygen demand and chemical oxygen demand reduction, as well as the nutrients removal in the bathroom greywater sample. The optimum loading capacity of Microalgae Botryococcus sp alginate beads were determined. The optimum loading capacity of Microalgae Botryococcus sp alginate beads for all parameters and nutrients removal was obtained, which is 600 gram, because when the 800 gram were conducted, the efficiency of the 800 gram loading capacity was reduced. Thus, the optimum loading capacity for the Microalgae Botryococcus alginate beads is the 600 gram. So next, based on the graph, it shows that the experimental data was not in line with the pseudo second order kinetic model. This can conclude that the pseudo second order kinetic model that is not quite suitable for the absorption analysis of the microalgae. The previous research also has found that the pseudo second order is not suitable for the analysis of microalgae. Nonetheless, all the parameters and nutrients that were checked in this study show the constant reduction throughout the phycoremediation process. So as for the conclusion, the efficiency of Microalgae Botryococcus sp alginate beads in the reduction of parameters and nutrient removal is achieved in this study. Next, the optimum loading capacity of the Microalgae Botryococcus alginate bead is obtained, which is 600 grams. Lastly, it can be concluded that the pseudo-second order kinetic model is not a suitable absorption kinetic to use in the analysis of microalgae. There are some kinetic models that is more suitable for this absorption of microalgae, which are the Langmuir and Frodelic isotope. So that is all from me. Thank you.